Good day, everyone. Welcome to Anti-Venom Snake Biotechnology. Biotechnology, known as agri-tech, that involves scientific tools and techniques. It is actually ensuring the genetic, molecular, and vaccines, and even the tissue culture, as well as the markers and diagnostics, which typically it is utilizes the living cells and cellular materials just to create a pharmaceutical and products benefits to society. These venom snakes are actually those species that are suborder serpents, meaning to say they are capable of producing venom which used for killing of prey and defenses. But these snakes are actually harmful for a fact that they contain short life for human span. They're always brought fears and phobia to both human and animals, which often be untrusted and bring wide risk in losing lives. These snakes are actually had good sides for somehow reason that they contain for introducing medicines, but they actually have these complex mixtures of neurotoxic, cardiotoxic, cytotoxic, nerve growth factor, lectins, and many other different enzymes and proteins which resulted to kill animals and humans. In overview, these snakes had tremendous effect in losing lives, which counted to 5.4 million people estimated beaten each year. That comprises the 2.7 million envenomings that largely range to 81,000 to 138,000 people die each year. Thus, the center point for topmost venomous snake in the Philippines is actually known as Philippine king cobra that fearfully kills people with three major ways attack to human body, which are the paralysis of the heart and respiratory, as well as the lungs.
Largest venomous snake, which is the king cobra, can found in jungles from India to Southeast Asia, including the Philippines. The amount of venom produced by each of these species varies, although it typically ranges from 90 to 100 milligrams per bite. The venom of Philippine cobra is so lethal that it can target the respiratory system of the body in minutes, paralyzing the breathing muscles. Neurotoxicity was the most common clinical characteristics in 38 of the cases. In 19 victims, respiratory paralysis developed quickly, with apnea occurring in 3 cases within 30 minutes of the bite. There were 2 deaths, both of which occurred in patients who were already dead when they arrived at the hospital. The patients developed necrosis, while 14 people who had systemic symptoms did not have any local swelling. There was no cardiotoxicity or reliable nonspecific indications of envenoming. Bite wounds from the Philippine cobra have a specific clinical profile that includes acute neurotoxicity with rapid onset and minimal local tissue damage.
Statement of the problem. For this animal biotechnology proposal, the snake venom biotechnology, we have identified the gaps and the questions between the current state and the desired state. And these are the following questions. For question number 1, how does the venom react to the human body in other animal species? Number 2, what are the benefits from this biotechnology? And number 3, what are the effects to the snake, to the human, and to the environment as well?
In addition, how this project relevant to society and what and how goodly relationship towards snake and human.
The physical description of cobra. The king cobra's average size is 10 to 12 feet, but can reach up to 18 feet. The full-grown king cobra is yellow, green, brown, or black. There are usually yellowish or white crossbars or chevrons on its body. The throat is light yellow or cream colored. The juveniles are jet black, yellow, or white crossbars on the body and tail. And for similar similar crossbars on the head. The king cobra is regarded as a fierce and aggressive snake. Its length and size give it an awesome appearance. Moreover, the king cobra was ranked as the top one most venomous and deadly snake here in the Philippines. Now, how king cobra is alarming to human? Patients bitten by the king cobra will possibly die after 20 minutes or 10 to 20 minutes. There are series of recorded deaths of king cobra bites. One of those was two farmers who died of king cobra bites, which was happened previously at Bansalan, Davao del Sur.
The plant kingdom provides an alternative to anti-snake venom. The medicinal plants have been used as folk or cultural medicines for treatment of the snake bites. Reliance on medicinal plants is primarily due to their safety, effectiveness, and cultural preferences. Globally, traditional healers are practicing herbal medicines to cure the snake envenomations. So there are a lot of medicinal plants available here in the Philippines like Cordia verbinacea, Tamarindus indica, Anacordium occidentale, the lupul acetate from roots of Hemidismus indicus, and lastly, the Strychnos nux vomica used by the tribals for snake bites that is reported to be the anti-inflammatory. So these are the existing solutions before that used to cure the ESV.
Evolution of bite of king cobra of our recent time around in the Philippines under the NAHA Philippines or RITM. The Research Institute for the Tropical Medicines, they are producer and distributor in the Philippines since 1960s. For the purified cobra anti-venin or FICAV, the only known effectiveness antidote for the snake bites, especially those caused by the Philippines cobra. Their venom is mainly neurotoxic, but it can also harm to the body tissue, to the blood cells of human. FICAV was only moderately effectiveness in neutralizing the toxicity of the venoms. Its potency was even lower against the heterospecific. The importance of this anti-venom therapy effectiveness and safety to the human, and to be recovered immediately those humans that victims of the bites of the king cobra.
Proposed solution, so we will use another method for making anti-venom. This method will have a good benefits both on humans and snake. So we will use a biotechnology technique, the germline gene therapy, and the Crispr-Cas9. We will insert or inject liquid on the snake that will contradict the venom, and after mating, the mutated ones will carry the antivenom that will help us humans to withstand the venom of the snake. So this method is really important because the antivenom don't last long and it's very expensive. Not many people here in Philippines can afford it. With this method, we can help our country, especially on snake-prone areas. The most important on this method is we don't have to be afraid of the venomous snake anymore. That's the reason why we kill venomous snakes. It's because they carry a deadly venom that can potentially kill us humans or animals, and their number is decreasing. That the Philippine king cobra is on endangered species list. With this method, we don't have to do that anymore, and we will leave the venomous snake alone because the snake has a very important role in nature.
Means and its goods. In this biotechnology solution, there is always advantages and disadvantages. And for the advantages is to improve the quality and health of the snake, to improve disease resistance, and to minimize the fear of humans towards the snakes. And for the disadvantages, the inserted gene has multiple functions, having low survival rate of the snakes, and lastly, disturb the natural being of the snakes.
Now, in having neither making the main theme culture or cultivation of this, must adhere to such considering that of course you have to propose a checklist as your lineup from subjects, pattern, uses, and biotechnology techniques to be applied and given efficiently. Thus, it categorized into two media or ways to process and perform the said first and main product and stage to pursue, and that involves neither came from spices and flour as herbs, which naturally consist essential elements productively helpful and effective to both human and animal. The two used media are defined anti-venom media that is actually had this quantity of ingredients and elements indicated for anti-venom that consist of antitoxins, Vitamins consist of zinc, iron, B1, B6, B12, and even macronutrients. Now, ASV cultivation can be 80 times more efficient and effective in a minute than traditional cure. Now, there's lots of prominent and prior species that are prone for that cases and painful disaster to human state, which compose of waggler spit viper, king cobra, yellow lipid sea crate, or just to have the family of cobras, coral snakes, viper, and sea snakes. Now, ASV cultivation is actually an unusual nature system that is adapted several spices extract mixed well, and it is contained or presumed in high ventilated isolation. Now the other one, media that composed is complex media, which it is wide materials of spices that consist of garlic, onions, ginger, eucalyptus, or even a virgin oil. It is also the media that literally choose based on its capacity, essential elements to contain, and helpful to human system. That natural virgin oil from olives makes a well-defined liquid syrup type of antivenom and to makes the medium. Now, natural virgin oil, or NVO, is a flexible element consisting 9 to 10 essential, known large and various amount of desirable compound. For ABR, or antivenom beneficial reactors, the culture is actually a directly mixed variable extract contains spices and herbal oil that had better contain elements yielding for effective input and massive reactant to antivenom or to venom that must be heated and enrich the accurate extract content of spices and herbs to outcast the significant trace elements and further natural compound. Now there were two techniques to be applied, and that is CRISPR-Cas9 method for the targeted gene and germline gene editing for the preferable of passing antivenom inserted.
Crispr or Cas9 is a technology for inserting sequence a specific position in a genome. This allows for specific editing deletion or exchange of sequence in organism, where homologous recombination is not straightforward. So in general, it can be used to efficiently engineer genomes. This virtually reduces the time to create transgenic organism. For instance, with alignment of our topic, we have injectable liquid to the snake to break a particular gene in order to reduce our fear of the risk that will pose to us if we are bitten to the side effects of snake venom. Moreover, it is a very powerful tool in genetic research and synthetic biology. So in a way, it can blur the line between classical genetic engineering and synthetic biology, and greatly reduce the effort and permanently change the genetic setup of an organism.
Together with germline gene editing, the preferable of passing beneficial venom is now inserted in specific reproductive cells of both male and female venomous snake. That actually comprises the purely ABR liquid syrup. Thus, the collision of CRISPR-Cas9 method and germline gene would have wide assurance of positive possible outcome. Their natural sense and mean to human will changeably into good, thus wiping the decreasing rate lives of both human and animals, as well as positive interaction is simultaneously brought pleasant outcome to the whole nation, ages, and society, or even the community as well.
Our mission, vision, goal, and the provision of this plan scheme is to develop and create an efficient treating cure or solution against the venom of snakes without adverse effect to human health, to protect and consider the welfare of every human lives and wild animals such venomous snakes, to protect the habitat of venomous snakes, and consider the survival of humans by making efficient treating cure or solution medicine against the venom of snakes, to produce new efficient anti-venom with no adverse effects on human health of science and technology, where new ideas of efficient anti-venom of snakes distributed, flourish, and bloom to next generation. All activities shall be authorized by the government upon proper evaluation of this available information or scientific data showing that the activity is, or for what a purpose, that is not destructive and without adverse or unwholesome effects.
There were several processes to make an antivenom, and that comprises 6 steps: milking the venom, cooling down and labeling, choosing an animal for immunization, immunizing, purifying, and human use. For the executive summary of regulations or guidelines to apply the submit proposal comprises 4. Prepare a checklist. Consider the objective, mission, vision, and goals, nature and background researches of the study or the subject, experimental procedures, and the last one is release, reuse, and readmit.
Regulatory compliance, on the other hand, is a set of rules that every must have to consider or must have to follow in order to comply with the law. And relevance to our topic, we must have to ensure that we respect animals' worth as regardless with their utility value, and that we investigate whether it is possible to limit the number of animals used in the experiment and to preserve the experiment scientific quality, the outcomes of the experiments, and the significance of the findings.